Fungi are among the most fascinating and misunderstood organisms on Earth. They feed us, heal us, and even threaten us. Some species can break down pollutants, while others can kill us with the slightest touch. They exist in food, medicine, pop culture, and even in our own bodies. And yet, despite their immense impact on life as we know it, fungi remain one of the least studied and most overlooked domains of biology. But for Dr. James Scott, fungi are not just an academic interest, they are his life's work. As a mycologist and public health scientist, Dr. Scott has spent decades studying fungi's role in medicine, disease, and the environment. He has advised poison control centers, uncovered new fungal species, and helped identify fungi that shape everything from antibiotics to whiskey distilleries. However, today's conversation is about more than just the beauty and mystery of fungi. It's about a crisis that could erase a century of scientific progress. Right now, Canada is at risk of losing the largest and most important research archive of public health relevant fungi in the Western Hemisphere, one of only 2 such collections in the whole world. The University of Alberta Microfungus Collection and Herbarium, currently housed in Toronto, is a scientific treasure trove containing over 12,000 unique fungal specimens, many of which exist nowhere else on Earth. These fungi have played a role in medicine, agriculture, environmental science, and they hold the secrets that could lead to the next great medical breakthrough, just like penicillin, which was discovered by Fleming in a forgotten fungal culture. And yet, due to a lack of funding, this priceless biobank faces imminent closure, leaving its future uncertain. Some of these fungi could be sent overseas, restricted from Canadian scientists. Others could be lost forever, simply discarded as if they had no value at all. In this episode, Dr. Scott makes the case for why this collection must be saved, why government and private donors should step up, and why the loss of this archive is a crisis for all science, not just mycologists. Dr. Scott and I also explore the wider world of fungi, from the role of mycelial consciousness, panspermia, as well as the rise of fungal infections and the incredible medicinal potential of these organisms. This is a story of scientific discovery, government bureaucracy, and the fight to preserve something that could change the future of medicine. Join me in vivo with Dr. James Scott on the fungal archive the world cannot afford to lose. Is there any other organism that evokes such a diverse set of reactions? There's gastronomical interest because they're tasty. There is trepidation and fear because they can be toxic. Right? There's reverence because of medicinal properties, psychedelic properties. It seems to permeate popular culture everywhere. It pops up in children's cartoons, movies, video games, everywhere, right? So what do you think is the source? Why are people so fascinated with mushrooms and fungi? There's, in so many historical and cultural contexts. What is it with humans and mushrooms? What's the connection there? Why are we so fascinated?
Well, they're strange things. They're really unusual things. They're about as close to aliens as we can get on this planet.
Right.
They do all kinds of things that are just fundamentally unfamiliar. They reproduce in ways that we can't really even comprehend.
Right.
But they've also long been kind of estranged from the public consciousness.
Yeah.
And so I don't know what it is about the last maybe decade or so that people have started to become more enthusiastic about them. I've seen this. Like when I was an undergraduate student, I got involved with the Mycological Society of Toronto, which is the amateur mushroom group in Toronto. And at the time, I think it had about 50 members, and the median age was probably about 60.
These are academics and non-academics alike?
These are retired accountants and stuff.
Okay.
People who are just enthusiastic about mushrooms. A lot, from Eastern European heritage and coming from cultures basically where, they already have some kind of appreciation of mushrooms. But, basically people wanting to get together and go out in the field and collect mushrooms, talk about them, that kind of thing. So about 60 or so members in the 1990s. In the last 8 years, the membership has gone to over 1,000.
Wow!
So there's for sure something that's bringing attention now on these organisms that people have long overlooked.
Right. You think that obviously has to do something with popular culture. I mean, but this is over the last 20 years, you're saying, right? Because recently, if I had to point to something, I mean, for example, there was that really good Netflix documentary, right? There's talk, you see it in podcasts and that sort of thing. There's obviously a fascination with psychedelics and that sort of thing as well. Paul Stamets, popular mycologist, not necessarily an academic, but has sort of pushed the envelope on that. But I think the first thing that you said there is that they're so strange, right? We don't quite understand what they're doing, or I should say lay people don't understand or completely comprehend what they're doing, but there's something there that we connect with. You think it's food? Do you think it's food that sort of the gateway as a food?
I think in a modern way, probably food and this idea of eating locally and foraging for wild foods. I think fungi probably benefited significantly from that. But I think it's more than that. When I have gone out on field trips, for example, collecting mushrooms with amateurs, there's only about half of the people that go on those trips that are actually interested in eating the mushrooms.
Oh, is that right?
The other half they're just interested in the mushrooms on their own. They want to know what they are. They want to know what they do in nature. Right. They want to take pictures of them and, understand sort of what their stories are, what their role is in nature. So I think it's more than just food, but for sure, food has this idea of foraging for wild foods has certainly drawn a lot of attention.
Well, I mean, even if you think about your grocery store in the last 10 years, it used to just be white button mushrooms and shiitake, but now you've got a whole variety of mushrooms that I wouldn't even know how to pronounce some of them, right? So that would be, I guess, an example of that. But I think, it's a beautiful way to get out. Right? You're getting out there, you're walking in nature, you're picking, you're foraging, you're taking pictures, like you said. You get to learn about biology hands-on with experts such as yourself if you're part of a group. That's a beautiful thing. How did you personally get interested in mycology? Because now you're an academic and you're studying this on a level that 99.9% of people on the planet don't study it at your level. How did you go from foraging to where you are now?
So there's a long story and a short story. So I'll give you the shorter version of the story. So as an undergraduate in the mid 80s, I was kind of this disaffected student who wanted to study science but didn't really know what to study. And in my very first biology course, I had a professor who was doing his little piece of a much larger course. And he was talking about fungi, and he told a couple of stories about fungi. And the thing that struck me when he talked about fungi in his class was that he pointed out that there are a couple of species, and he gave examples that have been known for at least 150 years or so. You could go out into the field, and if you found the substrate and you saw the little orange dot on the thing, you could be pretty much guaranteed that was the fungus. But he said that but nothing else basically is known about it. No one knows how it gets from this substrate to the next substrate.
Right.
No one knows sort of how it interacts with other things. And his challenge to us was that, if you wanted to spend a week of your time and go out and, lay on your belly and watch one of these things and figure out maybe what insects visited it or something, then you could work out its life cycle and you would then be the world expert on that thing.
Right.
And as a young student, it was really the first time that I saw the limits of knowledge. And they were much closer than I expected.
Right.
So I thought,
and accessible.
I thought, to hell with undergraduate physics.
Right.
I don't need to do an undergraduate physics course. This is something that I can do, and I can go out and do this now.
Try to get your hands on a particle accelerator.
Basically. So, so anyway, I took I think 4 more undergraduate courses with him. Then after I finished my bachelor's degree, I joined his lab as a lab technician for 4 years. Then I went on as a graduate student under his supervision. Ultimately, after I finished my PhD, I joined the faculty alongside of him, and I gave the remarks at his retirement. So all from that one
life cycle,
that one introductory biology lecture that just it completely changed my life, but also sort of my outlook on science.
Interesting. And how has your journey in academia affected your outlook on fungi and mushrooms in general? Like when you walk into the grocery store, what are you thinking?
Well, I never really liked mushrooms that much.
Oh, interesting.
And for a long time, I don't mind mushrooms on pizza or something, but they weren't really my favorite things. Over time, I've gotten more into mushrooms, and I will go out and forage for mushrooms. I'm quite good at identifying mushrooms. I've, for the last 20 years, been the consultant to the Ontario Poison Centre on mushroom poisonings.
Yep.
So I've kept relatively proficient on mushroom identification, whether they're whole mushrooms or chewed up pieces or whatever.
Right.
But so I will go out and forage for them, and I've gotten more interested in eating them. But I was one of those people who was mainly just interested in understanding what their lives were. Their secret lives. They're sitting there doing these things that we don't know anything about, and
only the tiniest of portions we can see, right? Explain to our listeners, a mushroom is just a small part of the whole organism. It's a fruiting body. So, most of it, correct me if I'm wrong, is invisible in the sense that it's underground.
Yeah, the mushrooms are like the apples on the tree.
Right.
Right. And if you see a mushroom and you dig down in the leaf litter around it, you'll see maybe about 2 or 3 centimeters down this mat of white fibrous material, which is that's the vegetative body of the fungus, and it ramifies through the soil. Many of those fungi, depending what the species is, will have interactions with the roots of trees where they form these partnerships that are key to the ability of the tree to survive in that habitat. Like, for example, a lot of northern forests, the soils are so depleted in nitrogen and phosphorus that the tree, without using that fungal partnership as a way to harvest those nutrients, wouldn't be able to survive on its own. So those kind of partnerships are really, really important. But to your point, the mushroom fruiting body that's above the ground is just a tiny little piece of the whole fungus that's there.
So obviously, maybe not obvious to everybody, but all mushrooms are fungi, but not all fungi fruit into mushroom.
Correct. Yeah. It's only a relatively small set of fungi that are in that group of things that we call mushrooms or mushroom-like fungi. Many of the things that I work on are sort of the sister group to that, that are relatives of what we call the cup fungi. And those are much smaller, tiny little things. If we look in the plant sitting here, we could probably dig around and find a couple of those. In fact, some of the white stuff on the top of the plant soil is likely an asexual stage of one of those cup fungi, which we would call
CUP.
Yeah, which we would call a mold.
Interesting.
So a lot of my work has been on the very small ones, the microfungi, the molds, rather than mushrooms.
And what utility do these cup fungi serve in terms of research? Do they have medicinal properties, or what purpose do they serve?
Well, I think they serve as a focus of interest more than anything else, right? They are the objects of these interesting biological puzzles, because many of them, like we think of molds as being these sort of interchangeable things that grow on stuff in the refrigerator or they grow on our shower curtains and things like that. But really, there are thousands of species, different species of these things, and each of them is very habitat specific. So, for example, if you get an orange that goes moldy on your kitchen counter, the fungus that's causing that blue fuzz to appear on the orange is probably a thing called Penicillium digitatum. But if you took a little bit of that and you stuck it on a needle into an apple, it would never grow on an apple.
Interesting.
A different species of penicillium grows on an apple, Penicillium expansum.
Okay. And
when it grows on the apple, it produces a toxin called patulin that causes people to get sick when they consume cider that's at fall fairs. It's been contaminated by moldy apples. So So each one of these fungi, like we think of them as being these sort of interchangeable generic things, but they're very, very specific. Many of them are specialists.
Mm. And I'm just curious, in your time doing your research in your foraging and your exploring out in the woods and whatnot, have you yourself identified anything unique?
Oh, for sure. Yeah.
Yeah.
We all do that. Yeah.
Wonderful. What will be the most interesting thing that you've come across uniquely?
Probably the most interesting thing was a fungus that I ran into on porcupine dung. So This was a collection. The 4 years that I spent as a lab technician was funded through the National Institutes of Health to a grant jointly held by my PhD advisor and a natural product scientist at University of Iowa. And the idea was to look at these habitats that are very nutrient-rich habitats, but that don't last very long, because those are the sorts of habitats that favor chemical competition between organisms to be able to access the substrate. So the habitat that we were looking at was herbivore dung.
Okay.
So we're looking at droppings of, moose.
Which doesn't last long.
No, it breaks down, and it's as soon as the droppings are dropped in the forest, the droppings are these rich packets of nitrogen that are dropped in this landscape that's completely depleted of nitrogen.
Right.
So there's a lot of things that really want to access that. So the 4 years that I spent, I collected a lot of droppings of various herbivores and would take them back to the laboratory and keep them in little moist chambers. Yeah. And over the weeks that ensued, I would look at those under the microscope and pick off fungi that emerged and get those into pure culture and identify them. So one collection that I looked at of porcupine dung had an interesting kind of little coily fungus on it that I got it out into pure culture, and it resembled a couple of things that were known. I was able to kind of figure that much out. But then when I went deeper, I really couldn't come up with a name for it. There was always something that was wrong. Like its spores were, football shaped when they should have been round, or things like that just didn't really fit into any of the existing categories. And we determined that it was not just a new species, but a new genus, which we named Polytolpa.
For specific reasons?
Well, many skeins of yarn, because it was
okay.
So, and Polytolpa hystricus after the porcupine, so in the years that have passed, others have looked at that fungus, and even though I was quite young and inexperienced when I described the thing, it turns out that it is, in fact, it's a distinct genus. Only one species is known, only Polytolpa hystricus. And so far it's only known from one collection, but it's the only non-pathogenic ancestor of the group that contains all of the most serious human pathogenic fungi.
Interesting.
So it's actually been a fairly important genus in terms of understanding the origins of pathogenicity in that group of
right.
of fungi.
And what makes the others pathogenic?
Yeah. So that's probably one of the more interesting things that I've run across, and it was partly by accident, partly by just me wanting to see what the thing was and dig down and find an answer to the question and persist. And I think I was fortunate enough to have nailed it, but I could have easily been wrong. It could have been something that someone else had described and I just didn't know. But so it was kind of lucky.
What I find interesting about your sort of your path, your trajectory is you'd say you're an expert, obviously, in pathogenic fungi. Most people, I don't think of fungi in terms of pathogenicity. They might think of it in terms of toxicity, which I don't know is the same. Would you consider, is that the same thing?
Well, I guess it's not. Toxicity is kind of a form of pathogenicity,
right? But it's not a disease.
There are things that that could cause an infection that in doing so would produce toxins that would elicit responses in their host.
Right.
There are other things that aren't able to cause infections, but they produce toxins, like the fungus that I mentioned on the apple, right?
Right.
It can't grow at body temperature, so it's never going to cause an infection in a human. But certainly the toxin patulin that it produces in the apple will make you really sick, like seriously, gravely sick if you ingest a lot of it.
How common is that? I mean, considering that you advised the Ontario Poison...
The Poison Center. But they just deal with mushroom ingestions.
That's it.
Yeah. They deal with mushroom ingestions. They deal with, acetaminophen overdoses. They deal with all kinds of things, right? Children who scavenge bottles underneath the sink in the kitchen.
That's right. So in terms of, let's say, because I've never really sat down and thought about pathogenic mushrooms. I think most people, as I mentioned, they think of mushrooms in terms of food, and then they think of the pop culture psychedelic. But pathogenic mushrooms, where we sit here today in Ontario, South Ontario, how common are pathogenic mushrooms, or fungi, I should say, in the wild at home? So
there are very, very few mushrooms that could cause like an infective disease in humans, but there are some. Like there's a little wood rot fungus that's it's just a tiny, tiny little shelf fungus that causes regularly lung infections and infections of other organs in humans, usually where there's some kind of pre-existing susceptibility like immune suppression and that sort of thing. But most of the fungi that cause human diseases aren't on the mushroom side. They're on the cup fungi side or the mold side. Right. And their relatives, the yeasts, which are, they're sort of interspersed. Right. Yeasts are one of those things that not all yeasts are related to all other yeasts. Right. It's something that turns out to be a convenient solution to a problem of growing in fluid environments, and multiple groups of fungi independently have given rise to yeast-like forms. So yeasts sort of pervade the fungal phylogeny. But most of the things that cause infective human diseases are yeasts or filamentous fungi that aren't mushrooms.
Understood. You mentioned in your early work pure cultures. How easy, or it's something that I've done and a lot of scientists do with bacteria. How easy or difficult is it to generate or to come up with a pure culture of fungi?
It's usually pretty easy.
Under the microscope?
That's where you start. Just working very carefully and picking off very small amounts. Like usually when we'll start from natural materials, we will use growth media in petri dishes that contain antibiotics to inhibit bacteria, sometimes some select antifungal agents that will not necessarily kill fungi, but slow down their growth.
Okay.
So we'll transfer a little bit of material to that petri dish and then wait for something to grow out. As soon as we see something growing out, if it looks like it's just one thing, then we'll transfer it to another petri dish, and a couple of steps like that, and you can usually get a pretty confidently clean culture. You can certainly verify it by looking at it under the microscope, and usually alongside that, getting a couple of DNA sequences from it just to make sure that you're
I was just going to get to that. Yeah. I would imagine that whole genome sequencing, or sequencing not just on the whole genome.
Just one or two genes, or even sections of genes.
Right. So, okay, understood. So I guess this would be a good point to bring up quite a serious issue with UAMH, which is the University of Alberta Microfungal Collection and Herbarium. It's an ironic name in the sense that it's got Alberta in it, but it's located here in Toronto, of which you are the director. And the name sort of suggests that it's been under threat before. But this is the largest archive or collection of medicine-relevant fungi in the Western Hemisphere, one of only 2 such collections in the world, as I understand it. And it's at threat of being closed because it's recently lost funding. And just to put things in context, you've got about 12,000 specimens there, most of which are uniquely held there, nowhere else in the world, which is priceless and wholly valuable. I mean, you only need to be reminded of Fleming and his serendipitous discovery of penicillin in fungus to sort of, think about the secrets that these specimens might hold, right? So what I wanted to do today was hopefully get the attention of some decision makers that might have it in their means to prevent this from being closed. Moved out of Canada, worst case scenario, specimens end up in the trash, unfortunately. So I'd love to get their attention. Why should people, mycologists, and also lay people who are not mycologists, because this is valuable for the whole mycology community worldwide. Why should people care?
Well, there are a lot of things in the laboratory that require you to either be able to archive material that you're working on to provide other scientists an ability to verify your work
right
down the line, or build on that work, or for you to be able to access species of certain organisms in order to do work with them.
Right.
And if you think about this from the standpoint of a chemist,
mm-hmm.
If I'm a chemist, I can open up a scientific catalog and I can order, magnesium sulfate.
Right.
And then a couple of days later, a jar of magnesium sulfate comes, and I'm all good. I can do my experiments. With biology, you can't just as easily open up a catalog
Correct.
and order like this organism or that organism. Maybe for a couple of very common things you can do that. But if you get outside of those common things, then you just can't do that. There are very, very few places in the world that have these biobanks of these very richly diverse biobanks of organisms that sort of go beyond the one or two things that might be the most common things that you'd find in a high school laboratory, for example.
Right.
So the reasons I think that it's valuable are, first and foremost, if you or I are working on an organism, so say we want to do something with this plant and either understand why this plant is growing much better than the other plant or something like that, and we isolate some fungi and do some studies and say, oh, well, when we use this fungus, the plant grows really well and whatever. We want to publish that, but typically journal editors will require you to deposit strains of the organisms that you've worked on and described or added information about in your paper, so that other scientists can come along and verify your work or go further with it. They may want to sequence the whole genome of that strain that you identified or obtained from a plant or whatever. So being able to archive these kinds of materials and make them available to scientists for further work is a really key thing. And this biobank and others like it are the places that you do that kind of thing. And then likewise, if you want to do some kind of test, like if you are running a medical laboratory and you want to do drug susceptibility testing of fungi or bacteria for that matter, you need to be able to obtain specific strains so that you can benchmark the performance of those tests against those strains, that everyone uses those same strains.
That you're certain it's the right strain.
So then you're able to validate your testing procedure. And so that's the other kind of thing that we provide for a fee is access to those kinds of materials as well. So even though these are very small things and the cost per strain is relatively small, these are very little pieces upon which very large research projects can be built. Mm-hmm. And without them, it becomes very difficult to build those kinds of projects.
I would say almost impossible. Now, this is, as I mentioned, not the first time, right? The collection existed at the University of Alberta. They lost funding there, and you had a hand in saving it and bringing it to Toronto. How does it feel to be in the same position again, but now in Toronto?
Well, this is not a new problem, and it wasn't really it wasn't a new problem in 2015 when I took on this collection. this is sustainable funding is an issue for not just our collection, but it's an issue for all kinds of microbial collections across the country and across the world. There are very few jurisdictions where governments and academic institutions have really invested in these kinds of resources. It never used to be the case. Like in sort of the boom days of the 1950s and 1960s, when people were doing a lot of drug discovery on wild collected material and building these repositories, there's quite a bit of money put into developing these and making sure that they were accessible. But things started to change maybe around the 80s or so, and it became the kind of thing that, especially with the rise of work on model systems, where you had fewer and fewer scientists who were looking at a diversity of different things.
Right.
They would focus on, maybe one or two experimental organisms. So that was kind of the early, I think, threat to these kinds of microbial resources, is when sort of studies of model systems really rose. Up until 2012, this collection and a couple of other collections, a very large collection of algae that's now at the University of Waterloo, and a collection of bacteriophage, which are viruses specific to bacteria at University of Laval. And our collection, they were funded through an NSERC program, Natural Sciences and Engineering Research Council of Canada program called the Major Facilities Access. And Major Facilities Access, it provided top-up money for these research platforms that multiple investigators would use. So things like an icebreaker that's in the north, that, people would use for collecting fish or collecting deep sea cores or whatever, but that no one investigator would be able to, afford the diesel, let alone the crew and all of these other kinds of things. So, but they would have user fees, and then the difference between what was collected in user fees and the operating costs would be paid through the major resources, it was called, or the NSERC program. Went through a couple of iterations. So in 2012, the government under Prime Minister Harper mothballed the program, and that left all of these research platforms, large research platforms, really unable to make up that gap. The icebreaker still went on because it was able to find some other ways. I think there was a cyclotron that was funded. It was able to get some other funding. Right. So the large users of that program were able to find other ways to sustain. I think there were some computing clusters as well that were funded through it. But the smaller groups, like these microbial culture collections, as far as I know, have not been able to find anything that's really replaced that.
Right. And
it's been like a year-to-year thing for those of us who are running these microbial biobanks to be able to keep nitrogen in our tanks to preserve these materials, and continue to send them out to researchers who request them, that sort of thing.
Yeah, I find it interesting because the archive itself is priceless, because you've got specimens there that some of them, many of which have not been clearly identified. They're sort of mysteries. So what mysteries they hold within them can be super valuable. We don't know. So for future scientists. And the maintenance, from what I understand, is, relatively speaking, not all that expensive. We're talking about, from what I read, $150,000 to $200,000 a year. Though the estimated value of what you have on hand is about $20 million. So in terms of hours spent and what's, just the value itself to maintain these, but you're talking about $200,000 a year to maintain this. It's not all that much money when we think about government budgets. So to me, there's a disconnect. You've got something that's relatively cheap to maintain, but extremely valuable. Why is it that these people in positions of influence don't seem to get that?
Partly because it's too small. It's, if you have something that you want to fund and you want to at the federal level go to Treasury Board with an ask,
right?
You'd get laughed at if you went
Isn't
to
that
Treasury
ironic?
Board.
It's too small. We won't fund it.
That's less than maybe $15 or $20 million.
That's completely
so in a strange
down.
way, it's actually easier to get money for large things. This basically needs one curatorial tech to operate it, a full-time person. And it needs nitrogen to be in the tanks, and then with the recovery,
liquid
so
nitrogen,
it's it's maybe $100,000 worth of salary, and then $20,000 or $30,000 worth of materials, and then whatever space, which I have so far been able to get from my dean. So it isn't really that expensive to run, but it's also one of those things that fits in this netherworld between research and service.
Yeah.
So, when I've written to politicians about this and said, hey, you guys should be funding this.
Yeah.
They say, oh, well, you can apply for a grant. You can apply for an NSERC grant, or you can apply for a CIHR or a CFI grant. And then, I look at those, probably as you do too, and these are all grant opportunities for hypothesis-driven research.
Right.
Well, this isn't hypothesis-driven research. This is, I have to put nitrogen in the tanks, and I have to send things out to other people doing research. So this is really a research service. So then I explain that to them, and they say, well, maybe you should consult with one of our innovation advisors. And then you look into the program, and innovation advisors support the private sector to do R&D around processes or products or things like that. And we're a nonprofit company that owns the biobank, and it's through a joint research agreement with the university. It's housed at the University of Toronto. So the innovation advisors want nothing to do with us because we're not looking at making profit.
Right.
So those opportunities aren't available either. So there are no opportunities to fund this kind of thing. And then the question is, well, increase the fees and operate this as a proper private sector business. And you can't really do that either, because we've got maybe 12,000 strains that require a lot of maintenance. It may be that over the lifetime of us having that strain, that we either only provide it once to someone or not at all.
Right.
But we still have to look after it and make sure that it's pure and keep it going.
Yes. It's almost as though you have an obligation to science present and future to maintain. Not almost. I mean, that is part of what's going on here.
Well, it's historic too,
exactly.
Right? Exactly. The oldest strains that we have in the collection are from the 1890s.
Wow.
So these are some of the very first microorganisms that were obtained in pure culture and maintained ever since.
And as I understand, you have some specimens of Flemings.
Yeah, we've got a specimen of Flemings. We've for the agents of human pathogenic disease or animal disease, we've got multiple representatives of some of the major pathogens obtained over a century at points before the development of the drugs that are now used to treat them.
Right.
So in this biobank, you can travel back in time and look at genetic diversity in these pathogens prior to the use of
their exposure to these agents. Yeah.
So it's a tremendously valuable thing. It's it's irreplaceable. Like, you can't go back in history and collect those things again.
Right. And I mean, one of the arguments is that you keep pathogenic bacteria or fungi to sort of be able to develop medications. How does that work? How is it that, just for people who might not understand how it works, how would you describe that? You've got specimens that may not have been described or cataloged, but you're hoping that in the future, they might be valuable in that sense to be able to produce medicines. What's that thinking? How does that thinking play out? Sure.
So, if in drug discovery, there's a certain amount of drug discovery that you can do with general kinds of organisms. You could have a panel of 3 or 4 or 5 or 10 things,
yes,
and then you could screen a bunch of different drug candidates against those in vitro assays and petri dish assays, for example,
Right.
and see, oh, well, my drug inhibits this one and this one and this one, but not that one and that one. But then if you want to look at using those drugs against specific agents, it's not enough to just look at one or two in vitro before justifying kind of the next level of animal trials.
Right.
You'd need to get a panel of maybe 50 or maybe 100 strains and look at the performance of your drug across those and see if the findings that you had on a smaller panel hold up in a larger set. So that's the kind of thing that would be able to provide as well.
You mentioned politicians. I know last year you reached out and wrote to our Premier Doug Ford, Vic Fideli. Have you heard back from them?
Yeah, I got a message from Vic.
And
Vic suggested that this was something for colleges and universities to consider.
Okay.
And I thought, it's sort of reasonable, and I can kind of understand him pointing this back to colleges and universities, just because the appearance of this is that it's coming from a university, and it's this idea of the biobank is sort of you think, okay, well, this is for research and for universities. In fact, over half of the strains that we send out go to the private sector. They don't go to people in academics.
Right.
Actually, it's only about a quarter of the strains that we send out, or between a quarter and a third, that go to academic laboratories. A bunch go to government public health laboratories. A bunch go to the private sector to, do all kinds of things. Everything from drug discovery, the kinds of things that we've been talking about, all the way to making myco leather.
Yeah, right. Yes.
Or cleaning up oil spills.
Yes.
There's a huge number of industrial applications for these kinds of strains. So we support the private sector to a much greater degree than we do the academic sector, which was why I wrote to Mr. Fideli in the first place, because it seemed like with Premier Ford's announcement of the $43 million or so for a biosciences initiative in Ontario, this seemed like it could actually be a fairly easy win for something like that, even if it's just a matter of putting a year or two of funding towards it for a couple $100,000. It's a nice checkbox. You can say, well, we did this.
Yes.
So that was my pitch to Minister Fideli. In hindsight, I really should have made the case more clearly about the basis for the support of the commercial sector and the fact that this wasn't really a solely academic enterprise.
So in your estimation, what is the ideal solution here?
I don't think that the government is really going to pick this up. Like I don't really see it. I don't think that this is a problem that's really limited to either our biobank or others like it, or even biobanks in general. I think there are a number of important research platforms that just don't currently have funding mechanisms. Whether that's biobanks or access to helium if you're a physicist, or a computer scientist working on your quantum computer.
Yep.
Or medical isotopes. There are a bunch of these kind of niche pieces that are really critical to supporting certain kinds of research programs that can be multi-million-dollar research programs. But if you can't access those relatively small resources, then you just can't do those grand pieces of research.
Right.
So I think there is recognition at the level of the federal government that that there needs to be some understanding nationwide about what those things are that we're reliant on that are struggling right now, and find better mechanisms to deal with them. So I do think that there will come a time, and it may be sooner than later, that the federal government will essentially rebuild a program like that NSERC program that I mentioned. I don't know that it's going to be enough time for us. So I'm not going to hold my breath. But I think this is a problem that affects not just us, but a bunch of others as well.
How much time do you have?
Well, I've basically been paying for this personally, right?
Yes.
So
I know. I didn't want to get into the books, but yeah,
it's kind of a misery, right?
Yeah.
But you do what you can, and I just it's. I feel so strongly about the value of this, and there are so few of us who do this kind of work that I do feel like I'm Frodo with the ring,
right?
Yeah, and
yeah.
I've got to protect that until I pass it off to either someone else or drop it in Mordor.
Yeah.
So when I took this on in 2015, I did so understanding that I would probably have to pay for a technician to run the thing. So I seconded a technician from my private company. So she's not contributing to the income of the company.
Right.
She's just costing. But that's great, and I understood that's how it was going to go. So I was sort of mentally prepared for it, and I'd committed, I guess, to myself that I'd give it a decade and try my best to pound the pavement and see if I could find a more sustainable funding strategy. And if at the end of that I still came up empty-handed, then I would have to pass it off to someone who could look after it. So
worst case scenario, where does it go?
So there's really only two places in the world that I think right now are sufficiently stable and well-funded to be able to look after this into the future. And both of those places, that's their benefit, but both of those places have different challenges. So the one is a similar biobank in the Netherlands, in Utrecht. And it's funded through the Dutch Royal Academy, and it's of a similar age to our biobank. It's around 100 plus years old. And I'm confident that they would be willing to take this on. The problem is that being part of the European Union, they're signatories to certain export restrictions around country of origin. And that would actually prevent the return of some of the material, actually a non-trivial amount of the material, back to Canadian researchers.
Right.
But at the same time, I think they would protect it into the future. The other collection is in China at the state Mycology laboratory.
The state key laboratory.
It's extraordinarily well funded. They're doing immensely valuable research in this area, and they're quite deeply resourced. And so I'm confident that group would also be able to integrate this collection and preserve it into the future. But it's really, at the whim of their government and our government, and the extent to which those governments get along,
right.
Whether or not we'd be able to access that material going to the future. Currently, we have no issues, but that could all change.
So I hate to boil this down to a elevator pitch, but if you had to come up with an elevator pitch, what would it be to anybody who's in those positions of decision-making power?
Well, I don't think that government is going to be the solution to this, really. Like, it just isn't. I think the solution is more likely to be through philanthropy.
Okay.
And if we could scrape together a couple $1 million and put it into an endowed fund, that would give us an ability to pay out annually a technician salary, maybe a little bump up for whoever faculty member, not necessarily me if I'm retired, is running it, just to make it, to provide a little bit of an incentive for a faculty member to be able to take this on and assume the little bit of additional work that it would mean. But I think if we think about this more through a philanthropic lens, there are a number of stories that this biobank has that I think would have potential value, particularly on biodiversity. So half or more of the strains in the collection are Canadian biodiversity. Lots represented from the north, lots of plant-associated fungi. We have one of the world's largest collections of fungal symbionts, root symbionts of orchids from not just Canada, like important threatened native orchid species, but also from global jurisdictions. We've got a large collection from Madagascar of these orchid mycorrhizal fungi. Importantly, the seeds of the orchids won't germinate unless the fungal partner is present. So for all of these botanists collecting these seeds on very expensive excursions to exotic places and then putting the seeds in Svalbard seed vault, those seeds are worthless unless they have our fungus.
Interesting.
So, and I could go through our collection and there are sort of clusters of things in the collection that have a really interesting narrative that I think would have appeal to the right group of philanthropists. And I think if we can pull together enough of those pieces, we might be able to build a whole. And if, my pitch is, if my university is willing to float me until then and commit some of their advancement resources to finding those philanthropic partnerships, then I think we could probably build it. But I think that has a much, much greater potential for success than looking to the government. The government's not going to help.
Right. Surprise. Well, so let me ask you this. What is the most prized specimen at UAMH?
I don't think there's just one.
Yeah. I don't imagine there's just one.
No. Well, I could stratify it. If I had to sort of pull the most important ones out if the building was collapsing or something.
Yeah, right.
The ones that would be the most important are the so-called type strains. And we have about 1,000 type strains. Probably about half of those are not replicated anywhere else. So a type strain is anytime you describe a new species, you need to deposit in a recognized place an individual of that species that is sort of like the platinum iridium cylinder of the species.
Okay.
So then any scientists in the future who want to come back and understand what that name means, they go back to your type specimen. So these type specimens turn out to be extremely important in anchoring and stabilizing nomenclatural concepts in fungi. And you might think, well, nomenclatural concepts in fungi, like when I say that, my students would glaze over. But these things can be really important. So for example, there's lots of legislation, federal legislation, provincial legislation, international legislation, trade agreements that have written into them quarantine regulations around names of organisms. Mm-hmm. We won't let wheat into the country or potatoes into the country if they have this organism or that organism. Well, those names are all based in that stability of nomenclature that's offered by the type. So if the type no longer exists, then the name becomes a doubtful name that can no longer be applied, and it has all kinds of unforeseen implications.
Domino effect.
In global trade.
Interesting.
So not just global trade, but also manufacturing. Like if you have a manufacturing process that's like in the US, an FDA approved manufacturing process for making, I don't know, this drug or that drug using a couple of microbial fermentation steps, then the name of the microbe that you use turns out to be really important because it's sort of codified in your process.
Right.
And you can't easily change it. So if the name becomes destabilized, then it can become a bit chaotic.
Interesting. And if those disappear, then clearly that becomes even more difficult.
They're not all as important, right?
Yeah, right.
It's the commercial ones, but the thing is, you never necessarily know when something is going to become commercially important. Like Fleming's strain, right?
Exactly.
When Fleming collected the thing, I suspect that he didn't really think it was likely to be that important.
Not at all.
He published it in a little note, and then he ignored it for many years after.
Right. Just to give some context, the first true antibiotic. We're talking about
Yeah.
penicillin coming from Fleming's strain of fungi.
Penicillium rubens.
That's right. How many of those might you have in your collection? Right?
Oh, probably about a dozen.
Yeah, no, but I mean, strains that might have some sort of medicinal property like an antibiotic.
There are, and we
Right.
have others that are used that have been used for other similar kinds of things, like
Right.
the fungus that was used to manufacture, still is used to manufacture cyclosporin, which was the first. Was originally discovered as an antimicrobial drug, but it was the thing that revolutionized organ transplantation by selectively suppressing the immune system and preventing host rejection. So, right.
It's just,
so that's an example of one that's in the collection as well.
It's just baffling to me. Well, you know, I guess it's baffling to you and I that you might even find yourself in this position because it's part of your everyday life, going to an archive or working with a model organism, whether it's cell line, which we were discussing earlier, is far more lucrative than fungi or fungi.
My major professor was from California, so I have all these West Coast pronunciations.
Fungi. But now lost my train of thought. But to scientists, it's clearly obvious, right? Because you rely on these archives for current and future research. So that's why it's baffling to me. But I suppose I could understand, you know, why a layperson might not see the value in it. But trust us when we say that there is a huge value, you know.
Yeah, there's an immense value in it.
Right. So I know. I hope, not just for the Canadian mycology, you know, group community, that you do not lose this, because it's clearly very, very valuable.
Well, we're still trying. We haven't given up. And I've had a number of conversations, important conversations within my university, and thanks to the attention that was drawn to this issue by the CBC. Like, I was really grateful to the CBC, and it surprised me actually that they even approached me on this. It just happened that someone, a journalist, reached out to me about an interview on The Current over a decision that was being made at the COP conference in Chile.
Yes.
And we chatted a little bit on the phone, and I said this was about biodiversity initiatives, and I said, you know, it's really ironic that you're calling me about this right now, because I'm in the middle of writing a letter to Vic Fideli about this culture collection that we're likely to lose. That's, you know, a huge amount of biodiversity. And he said, "Oh, would you mind sharing that letter with me?" And I said, "Sure, I'll send you a copy." So I sent it to Vic and copied it to the journalist, and it all went from there. It was not through any concerted effort. I wasn't specifically looking for publicity. I'd already kind of given up,
right?
And so I've been heartened that this news piece, the column that came later in the.
Was it Wired?
No.
Not CBC.
No. There was Wired. There was a print piece later.
Okay, and this is how I came across your story. Is somebody had posted it on X, the story from CBC, which I'll link in the show notes below. It's basically summarizes the problem, but thank goodness for that. Because you are getting attention for it, and hopefully it helps.
Yeah, we may have a solution.
Yeah.
It's I'm much more optimistic now than I was 6 months ago.
Well, that's good.
So
that's good to hear. So what do you think in terms of mycology now outside of UAMH? What is the biggest challenge for fungal research right now? In terms of just research questions, what are the questions that are being asked right now in mycology that are sort of top of mind?
Well, in many ways, fungal research was always something that was fairly easy to do on a low budget, just because it's very frontier research. You know, there's a lot you can do with a little,
right?
But the development of readily accessible tools for genomics and other kinds of sophisticated analyses, they've been democratized to the extent that you can do now really, really sophisticated stuff and it doesn't cost a huge amount of money. The thing, though, is that today, I think, and probably for the last 15 or 20 years, people have increasingly relied on the power of those techniques, using strains that they may have in their possession or a little bit of material at the expense of going out and collecting new material. So relatively few people have, over the last 20 or so years, have been going out to the field and collecting new material and looking at things in nature. And as soon as you sort of become disconnected with what these organisms do in nature, the sorts of questions that you ask start to kind of drift. They become sort of more theoretical. So I think what we're likely to see coming on is more of a realignment, like people appreciating that, yes, we have all these very sophisticated methods, but the methods aren't sort of the research in itself. You need to ask questions that are biologically relevant questions. So I think people increasingly are going to want to go back and look at these organisms and their habitats and start to kind of build bridges between the lab and the field. And I think that the extent of public interest in fungi and going out in the field and looking at it that we started talking about at the very beginning of this, I think we're going to see that reflected in researchers. I hope so, because I think that's something that's been lost as people have become enthusiastic, rightly so, about these new methods.
Yeah, that's interesting thing that happens. You get these silos, not exactly silos, but you start working on, I can speak to molecular biology in cancer lab, and every day somebody in some group in some lab sits down at a tissue culture hood, and that becomes their world. And you slowly start to focus on a petri dish, right? And that's it. But zooming out and recentering yourself in terms of mycology, like you said, going back out to nature, I think that would be a very interesting recentering or refocusing, and to see what comes of that would be very interesting.
It's because there's so much, it's so poorly studied.
Yeah.
That, there are lots and lots of organisms that we know from only one collection ever.
Yeah.
And nothing else is known about them. And, those, I think there's probably a lot of really interesting biology. There's probably a lot of stuff that we could utilize in other ways that's sort of hiding amid that biodiversity.
Yeah, it's interesting with mycology. When I'm thinking about it here, you've got in popular culture, with lay people, not scientists, it seems there's an inversion where you have so many people interested in it. But hearing from you, it's this very small group in terms of academics and researchers. Whereas if you think about cancer research, it's almost the other way around. I mean, most of what happens in molecular biology is either cancer, and there's tons of labs, tons of researchers, and very few lay people that are actually interested in that. So that's an interesting paradox between the two. And why that is, I don't know. Well, it's back to what we started with. It's just fascinating.
Yeah.
Right.
Well, but it's also frontier, right? And you can be basically an amateur.
Yes. There you go.
At the same time, a world expert.
There you go.
There's a guy in Europe who's the world expert on one particular genus of cup fungi. And during the day, he's a welder.
Right. Exactly.
But, he's an amateur, but he just has developed a world-class expertise, and he's the go-to guy for that kind of thing.
Sort of the way it used to be.
And in mycology, you can still do that, but in relatively few other domains of biology can you do that. I mean, maybe you could do it with insects to a certain extent.
Right. Now, the first one that comes to mind for me, a historical example, is Mendel. He was a priest.
Yeah.
And revolutionized what we know about inheritance, right? But I can't really think of rather a modern version of some layperson who has affected molecular genetics or cancer research in any appreciable way. That's the beautiful thing about mycology.
Yeah, it's nice, and it's very accessible, and it comes back to why I got into this in the first place.
And the popularity of people like Paul Stamets, right? I mean, I think he's probably had a hand in making it more popular, and is a good example of that. So, what are you working on now in terms of research projects?
I'm still doing a fair amount of work with the whiskey fungus.
Yes.
This is a fungus
The
that
black fungus.
grows around neighborhoods where there are spirit aging warehouses that emit ethanol vapor and
Right.
and cause changes in the biology of surfaces for a couple of kilometers around the warehouse. So we're still doing some work with the whiskey fungus, which is kind of interesting. Finishing off a couple of projects, starting a couple of other projects.
And you also run a private lab, Sporometrics, that focuses on what exactly?
Well, so I started it in 1993 when I was a graduate student, and it was kind of an accidental business. My graduate advisor was on sabbatical, and I happened to get a telephone call from a fellow at the federal government, Public Works and Government Services, who wanted some air samples from a public building analyzed. I said, well, I can't do that. My advisor is away. And so I hung up the phone and I thought that was it. And then a couple days later, this box of samples arrived. And I opened the samples and I thought, oh no, what do I do with this? So I thought, okay, well, I'll look at the samples and then I'll write up a little report and I'll send it and I'll say, don't ever do this again.
Right.
So I did that. And then another box arrived. And by the time my advisor was back from sabbatical, this guy at Public Works and Government Services had given my name to a couple of other people. And so this thing had started and I was at every point, I tried to just stop it. So when my advisor came back, I said, I think we need to sit down in your office and talk about something that happened while you were away. And then I told him and he said, oh, well, do you have time to do it? I said, I guess I do. He said, well, okay, I have no problem with that. So that's how I started it. And I needed to incorporate it because I had an NSERC grant at the time that prevented me from taking money from other federal sources.
Right.
So, I went to the student legal aid office and I said, I need to incorporate a company. They said, we only deal with landlord and tenant questions. And I said, well, are you sure? And they said, no, well, maybe we can find someone that could help you. So I got a couple of first or second year law students and together we did the incorporation. So it was very, it was kind of the sort of innovation that universities like to foster now.
Right.
But back then they wanted nothing to do with anybody's stuff.
Now, that's not the first time I've heard of somebody spinning out a company while their PI was on sabbatical. It's literally not the first time. Last person I interviewed was exactly the same thing. And now, Holoclera, now they've got the first gut worm-derived molecule in phase 1 trials. It's all because her PI was on sabbatical at the time. So take an opportunity when you see it. But so you work with workplace. What would be the term, exposure.
Yeah, well, that's probably about a third of the work the company does. It does a lot. We did an ISO 17025 accreditation maybe about 15 years ago, and then we did a Health Canada GMP and drug establishment license. So we do a lot of work with pharmaceutical testing, cosmetic testing. For kind of routine stuff, we do some contract research for a variety of things, but all kind of centered around microbes. We do a lot of specialized testing for specific microbes using molecular genetic methods and some other methods as well.
I bring that up because I'm curious, how common is exposure at the workplace or even at home to pathogenic fungi?
Pathogens, not as much. Things that are either allergenic or irritant or toxic, probably more common. There are certain kinds of workplaces that tend to have lots of those kinds of exposures. The thing that comes to mind is agriculture, for example. There are lots of crazy exposures in agriculture. Obviously, potential exposures to pesticides and things like that. Lots of potential for musculoskeletal injury, but also there's a lot of potential for bioaerosol exposures in a range of different kinds of agricultural workplaces. But then also things you don't think about. I did a study maybe about 20 years ago on machinists and metalworking fluids. There's metalworking fluids. They used to be neat oils. So these are the lubricants that are sprayed on parts when they're machined or milled or drilled or honed to cool the part, but also to wash away the bits of metal that are taken off and shaping the part. So neat oils were used, and these were oils that contained PCBs because they didn't polymerize. So they had really desirable characteristics, but machinists had really high rates of occupational cancer because they were exposed to these PCB-containing oils. So about 30 or 40 years ago, there was a move to replace these PCB-containing oils with emulsifiable concentrates. So oils that will emulsify in water and provide the same kinds of lubricant properties and coolant properties, but not be carcinogenic. And cancer rates in machinists went down precipitously, but asthma rates in machinists went way up because these machining fluids were really susceptible to overgrowth by bacteria and fungi. And so you had these exposures that were bioaerosol exposures that were not necessarily anticipated. And now technology has advanced to the point where there are different additives that are used in the machining fluids that, some of them can improve the situation. There's still all kinds of issues with exposures in machining fluids and bioaerosol. But I give that as one example of a kind of industrial workplace that you wouldn't necessarily suspect has bioaerosol cylinders, but it's got a lot.
Seems like the last place I suspect machinists, right? Now, different topic. Well, mycology still the same topic, but completely different end of the spectrum. What is your opinion? Have you read or studied anything about mycelial consciousness? Asking for a friend. Justin, that's you. I'd be remiss if I didn't ask. The idea of distributed sort of networking and intelligence in the mycelia underneath the ground, the way they communicate and even have ways of problem solving. It's an interesting theory, and there's especially on Twitter and X is a huge community centered around sort of trying to define consciousness and intelligence and applying it to outside of just us and everyday animals that we see, right? Is this something that you've ever looked into or thought about?
I've thought about it a little bit.
Yeah.
So it's difficult for us to really comprehend how these organisms that we call fungi exist, right? Because, like I said,
fundamental question.
They're just these organisms. They're aliens, really.
Yeah. And they're curious when
closest we can get to an alien on this planet, and they're not like us at all. They're these sort of weird colonial organisms where you've got the filamentous ones anyway, where you've got these cells that they're all kind of attached, but they're also kind of separate. You can cut off one end,
right,
and one side of it will go on and do its thing, and the other side of it will go on and do its thing. But you can't really do that to a mammal like a human or a blue whale or something. You've cut it in half, then
exactly.
Neither half is going to be very good. But you can certainly do it with fungi, but at the same time, those fungal cells, while they're connected, they do communicate. You could, and that this has been known for a long time. You can feed a little bit of a radio-labeled substance to one end of a filament here, and very rapidly you can get it detected
right
in the other end of the filament way over there. And so this ability of things to translocate and move around through this mycelial network, it's really well established, and it's one of the ways that causes filamentous fungi to be so successful in the environment is their ability to move things around,
right,
to traffic and nutrients, and do so efficiently. So taking that a step further and kind of exploring a mycelial network as a source of a kind of consciousness, I think it requires us to redefine what we mean as consciousness. Like, I don't expect us to be writing poetry,
right?
Although you can outfit a mycelial network with sensors that can detect electrical changes in electrical potential,
Yes, absolutely.
and you can convert that into something that's sort of like analogous to a brainwave.
Right.
Right?
Yeah.
What it means, I don't know. Is that wave pattern that you're revealing, is that somehow reflecting the response of the mycelial network to its environmental exposures, or changes in the environment, how it perceives the environment? Probably is, right? But not in a way that we can really interpret, and certainly not in a way that is reflective of consciousness as we think about it. But I think it requires us to really re-examine what we mean by consciousness, and I think it's difficult for us because we're sort of locked into our paradigms right now.
Right, exactly. And also you can educate us on this, the way they communicate with the trees that they might not inhabit. What would be the term that they
Well, they partner with.
Yeah. Yeah. They have the ability to communicate with trees.
They do, in some sense. What that communication consists of is hard to know. Like, the one thing that I am pretty confident about nature is that its rules are pretty brutal.
Yeah.
So I don't know that there's a lot of altruism in nature. We tend, as you're walking through the forest, you have this sense that it's all actually used by these altruistic things. Sure. But the ant's eye or the worm's eye view of it, it's this horrible, hostile environment where everything is trying to eat everything else and
Always, nonstop.
so I don't know that there's an awful lot of altruism in whatever that relationship is. I think that there have to be some mutual economies to it for it to be able to persist.
We're not race at the bottom.
But certainly it by being in that relationship, those organisms, their success for each of them is greatly enhanced. So there's a lot of value in it. I think now, coming back to this question of how can we think about them? A couple of nights ago I watched Carl Sagan's YouTube lecture on thinking about things in 4 dimensions.
Okay.
And it's a brilliant piece of science communication where he talks about, the 2D world, and then how the 3D world sort of would interact with the 2D world.
Right.
And then he uses that as a kind of metaphor or an analogy for how us in our 3D world can sort of perceive the 4D world.
Right.
So I think when we think about this sort of situation in the fungal world, it's very difficult for us to project ourselves there. Of course, it's one of those things that's so unfamiliar that I don't think we have any analogies to really work with it.
And there's interesting work being done with sort of applying what we know about that communication and problem solving, even onto neural nets and programming and AI, trying to learn from that and see how it can be applied to the computer world.
Yeah. Well, this had been done I think in the 70s or certainly in the 80s using slime molds.
Right.
To solve basically the traveling salesman problem, which is one of these famous classic computer science problems that if you try to solve it iteratively, it's a very intensive problem to solve if you do it step by step way. But the ability of a slime mold to essentially parallel process this problem,
Right.
It can do it very, very rapidly.
Right.
And so it kind of functions as an analog computer. And so, people have known that for a long time, but it's been one of those sort of curiosities. It's like the parlor trick of mycology that you show undergraduates how something like that can solve a problem, and then you're done with it and you say, "Wow, okay, isn't that interesting?" But it doesn't really go much further than that. But it does show that nature has an ability to do things that remain always very surprising.
Yeah, and seemingly difficult to us. Yeah, that is interesting, isn't it? Now, just back to toxic mushrooms and fungi. What would you say to somebody who would say you're crazy for going out foraging? You're going to pick the wrong mushroom. You're going to eat that and you're going to die. How likely is that? Should that be a barrier to entry to anybody who wants to go foraging?
No, I don't think so. But it's it's like anything. It's like it's like berries. There are lots of red berries at the forest.
True.
And some of those red berries are tasty, and others those red berries will make you sick. And there's probably even a few red berries that could put you in the grave. And how do you know? Well, there's two ways. You can eat it, or you can capitalize on the knowledge of the people who came before you who ate it or knew someone who did.
Right.
And the way that we pass on that knowledge is through identification.
Mm-hmm.
And we are able to, collectively build on knowledge as long as we have mechanisms to be able to consistently look at something and identify what it is. So if you learn how to identify these things and you know what they are, then you're able to capitalize on the knowledge of the people who came before and hopefully not make mistakes. So the price of entry really to doing that is learning how to identify things. There's another piece though that's important to understand, and that is that if you pull together a dozen mushroom field guides and you pick one or two species of mushroom and look them up in the field guides, for some of the really well-known edibles, confidently all of those field guides in your pile will say yes, it's edible. Some will say, oh, it's really good. Others will say, well, it's edible, it's okay, it's not as good as this mushroom, whatever. But for other mushrooms, you can look them up in one guide and it will say, oh, it's edible. Another guide may say, I wouldn't eat it. Another guide will say, oh, it's poison. So strangely, there's a surprising amount of disagreement between field guides on edibility for a lot of species of mushroom. So the conservative approach for interpreting that is if you've got one or two field guides and any one of those field guides says, oh, I wouldn't eat it, then my advice is don't eat it. If they all say, yep, it's edible, and if you're confident that you know what it is, and especially if you've consulted with someone who knows what they're doing, and you can learn from their experience, not just looking at pictures and books, because there are things that you can't really communicate as easily in pictures, right? But if you do sort of both, like if you do some book learning and then do a little bit of mentoring with someone who's skilled in that, then I think you can quite confidently identify those things, just like you can with plants.
Is there, at least in our area, if somebody is interested in something like that, a group that, I guess it was the group that you...
The Mycological Society of Toronto, MST.
And they travel outside of Toronto?
They sure do.
Fantastic.
They have... they've got members from all over the province. They've also got members out of province and some members in the U.S. They're part of the North American Mycological Association, which is the umbrella organization for all of the amateur clubs in North America. There's probably...
You were the director of that at one point.
No, I'm the scientific director of the Mycological Society of Toronto, but NAMA is the umbrella organization for the amateur clubs. There's probably about 300 across North America of these clubs, and each one of them has, from 60 members to maybe a couple thousand members. So there's a lot of folks that are part of that community in North America.
That's a wonderful thing. That must be a cool group of people.
I've only gone to a couple of their meetings. It's not like I interact with the local group here, but the North American group, it has annual meetings, and they do forays, they do lots of talks and that kind of thing. But it's not, mostly they're mushroom enthusiasts, right? They work on mushrooms, and like I said, I'm on the other side. I'm on the microfungus side.
Got a question for your hypothetical question. If you could secure unlimited amounts of funds for a research project, which question would you want to look into? What would you like to research into in your area?
To
To put you on the spot.
No, it's a really good question. I mean, as scientists, we always think about things like that.
Yeah.
I mean, we to the exclusion of other things, right? We're always sort of focused on these things that, oh, if only I could do this. I think it's so I would actually go back to working on one of the things that I started on, and that was working with those fungi that get on herbivore dung. And those systems, they're so interesting because they're life cycles that are very tight life cycles. They're often fungi that interact with the animal. So the fungal spores won't germinate unless they pass through the gut of the animal. Then they germinate on the pellet, and the fungus then has mechanisms to get to the material the plant eats, whether it flies with it on an insect or it hurls its spores specifically so that they adhere to plant leaves, then the animal grazes, and then the life cycle continues. Many of the fungi that are in that, their timing and their emergence, it's very intricately timed so that one will sort of begin its life cycle when another one ends. Some of them will be timed together so that they'll do something together. Surprisingly, not a lot is known about those little life cycles. Probably there's no commercial importance of this at all. Like, this is strictly a very, very academic question. But I think if I had unlimited funds, I would probably focus very narrowly, very academically on something that probably no one in the world is interested in at all. And it would be that. And just because it's one of those things that you'd never be able to get competitive funding for,
Right.
because you can't spin it in any way.
Right.
But the life cycles, I think, they're so interesting, and it would be really fun to work some of those life cycles out.
Makes you wonder how many research projects, if only they'd be funded, in lieu of not being able to spin out into something profitable.
Yeah,
And if there
there's nothing profitable or that at all.
but it must be sure of so many projects and questions hidden in that, that end of the spectrum that are just fascinating. The way that you just described it, sort of small, tight life cycles, we think we have it tough, right? But I guess opportunistic, extremely opportunistic, right? Something has to be bang on before they can continue their life cycle. It seems almost impossible. They persist.
Yeah, and there will be insects involved in a lot of them, or other arthropods, and bacteria can be involved. So they're multi-organismal symbiotic interactions that are basically unknown. A few folks have worked out some of them, but again, no importance whatsoever. But at the same time, almost every major scientific discovery in the history of science has come from basic research, where people have not been seeking to find that thing. It's relatively little transformative discovery that's arisen as a consequence of concerted effort to make that discovery. It's mostly just, Alexander Fleming finding penicillin,
Right.
inhibiting bacteria in his petri dish by accident.
What are your thoughts on sort of this way out there hypothesis or thesis that spores of mushrooms or fungi came from space, panspermia?
That doesn't really seem plausible to me. I think it's feasible that our common ancestor, in some form, maybe in the form of a few scraps of amino acids,
Right.
came from space. That wouldn't surprise me.
Right.
It's still, I don't know that it passes Occam's razor, but
That's a good way to put it.
in other words, I think there's an explanation that's a plausible explanation that requires fewer logical leaps.
Yeah.
But it's clear that all of the things living on Earth, even the really disparate things, the archaea and all of these very organisms, unusual organisms across the spectrum of life, that they all share a common ancestor. So, that ancestor may have benefited from some component of panspermia. It's possible.
Right.
Um,
I think that fungi get roped into this idea of panspermia simply because they're very hardy, right? They've been known to survive very extreme conditions in the spore state, I think.
It's mainly because they have cell walls, right?
Right.
Like fungi are in this netherworld between plants and animals. They're like plants because they have cell walls,
Right.
but they're like animals because they do animal kinds of things. They're much more closely related to animals. So
Right.
they're sort of like animals with cell walls. So they are
That's a good one.
the best of both worlds in a way. So they're very adapted to these very difficult kinds of environments. And they're probably one of the very, very earliest things to have emerged from the oceans.
Right.
And it's probably as a consequence of them that plants were able to emerge from the oceans, right? Through that kind of partnership.
Is there anywhere on Earth where fungi have not been found? The first thing that comes to mind, the Arctic or the
No, so there's a famous paper. Well, it should be famous. It's not famous. It's at least old, from the 1920s. And the second author is Charles Lindbergh, who flew a mission across the Arctic and took air samples out the window of his plane. And from these multiple air samples, it was actually a sophisticated study. In the high atmosphere, they recovered this huge diversity of fungal spores. It's great. They were doing this paper for the 1920s. They've got a photographic plate in it. You have pictures of the fungal spores that were found. And some of these are surprisingly big. They're 50, 60, 70 micron size spores that are floating up in the high atmosphere that, who knows exactly how they got there. I guess just dust disturbance and that kind of thing. But it shows you that it's possible for these things to move over vast geographic distances through the high atmosphere. And fungi, of course, they have cell walls. A lot of the cell walls of fungi are impregnated with melanins that will shield to some extent against ultraviolet radiation. So the ability of these things to move over very long distances, it hasn't necessarily been demonstrated, but at least it's plausible that a lot of that material that Lindbergh collected and others have since looked at, it probably contributed to genetic diversity across continents.
Sure. What about this fact? Can you corroborate this? I've read multiple times. I've seen it in textbooks that nearly every surface on Earth has some population of yeast, which of course is also a fungi, on it. Is this something that you can attest to?
It's probably true, right? Because the main yeast that we see in the built environment is a yeast genus called Malassezia, which is an oil-loving yeast. So it's a yeast that's not related to baker's yeast or brewer's yeast, or yeast that you'd get as a yeast infection.
Right.
It's yeast that's actually related to mushrooms.
Okay.
So it's a completely different group altogether, but it's specialized in living on the oily secretions of humans. So where you tend to find it on humans is on the oily spots, like on the forehead, face, neck, shoulders, chest, that kind of thing. The yeast normally doesn't do anything harmful. Occasionally, if it colonizes the very upper layers of skin, it can actually cause the skin to become a little bit darkened in that spot. Or alternatively, if you expose that skin to sunlight, the skin that's affected by the yeast won't tan. So it leaves sort of a white geometric pattern. So it's a disease. It's a superficial disease, but it's called tinea versicolor. The yeast also contributes to dandruff. or seborrheic dermatitis. But we're all of us covered in this yeast. Usually not just one species or one strain, usually like multiple strains. Like the yeast that you get on your forehead is going to be different than the one that you get on the back of your neck. That's a very, very specialized. So we're like this mega habitat for these things, and we shed them into the environment like anything. So if you look at the DNA, microbial DNA in house dust,
Right.
a lot of it is that yeast
Interesting.
that we contribute.
I never thought of it as it's coming from shedding.
It's coming from us. So yeah. So there will be other things as well. There are lots of wild yeasts around that are more closely related to brewer's yeasts.
Right.
They're
Just personally my favorite use.
many of those guys. Yeah.
They're just we were talking earlier about mycology as a niche field of study, or at least a underrepresented, or not a very large compared to some of the other biology sort of umbrellas. What would be your advice to students like a young Dr. Scott who are interested in mycology but are having a tough time finding a sort of imagining a path to being able to flourish ultimately as a mycologist, either professionally or academically? What would be your advice to a student who's interested in mycology, don't know where to go, don't know what to do, but they're interested in it?
So the piece of advice that I was given when I was considering going to graduate school for mycology was don't do it. You'll never get a job. And I said, well, but I want to do it. I don't really care if I get a job. Like, this is the one part of my life where I can sort of justify doing whatever I want. Like, when for the rest of my life am I going to be able to do this? So it was a very self-indulgent thing to go to graduate school and spend multiple years of my life working on something that didn't necessarily guarantee a job at the end of it, or guarantee a job, even have potential for a job right at the end of it. But it's what I wanted to do. Like, I knew that I had to do it. So I wasn't really thinking about a job. And a lot of the academic job that I've had has not been doing mycology. Like, a lot of my research specialty has been around bioaerosols, and I've probably published far more papers on bacteria and human microbiome. I was able to do a lot of that kind of work. I've published papers on viral aerosols. I've done fungal stuff as well, a little bit of fungal stuff, but most of my research hasn't really been in mycology. My graduate work was in mycology. My passion is still in mycology, and I found ways to weave mycology in, and sort of the paradigm of mycology has been reflected in other work that I've done. Like, I've tended to think about not just like single domains of organisms while I've worked on them, but I've sort of broadly considered biological systems, and fungi are part of those. So the advice that I would give is, don't do something because you want to do it. Don't do something because you think that you're confident that you're going to get a job at the end of it, because you may not, quite easily. Like, things can change really quickly. And I think it's more important that you get a foundation in something that gives you a framework to be able to do other things. And at the same time, I think it's important not to exclude yourself from things. Don't go to a job interview saying, oh, that's not what I do. This is the only thing that I do. Go to a job interview with the idea of, I can do that. That's totally what I do. I think studying anything can kind of prepare you for that, right? You don't necessarily, you're not locked into doing the same thing for the rest of your career. I've done a whole bunch of different things, and where I've needed support on the technical side, I've simply collaborated, and it's worked very well.
I think that's great advice. Closing question: if you had to dispel one myth about fungi, mushrooms, or molds, what would it be?
Don't eat moldy peanuts. Oh. Or don't buy peanut
This hurt.
peanuts or peanut butter in health food stores.
Okay.
Not to malign health food stores. They do lots of good work. But Skippy and Kraft are rigorously tested for aflatoxin.
Okay.
And peanuts from other sources are not.
Interesting.
And peanuts and peanut meal, when they become contaminated with Aspergillus flavus, which produces aflatoxin, it won't necessarily, it's very, very toxic acutely, but its long-term toxicity is the greater concern. And if you look at a global map of consumption of peanuts and peanut products and overlay that with a global map of the incidence of liver cancer, they match just about 1-for-1, with the exception of North America. And that's because our peanuts and peanut products are so regulated for the presence of aflatoxin. No amount is tolerable, but that's not true in other places in the world. So I would say avoid peanuts that haven't been scrutinized through that process. And if you're in any country other than a North American country, peanuts are probably not the stuff you want to eat.
Fantastic. Listen, Dr. Scott, that was awesome. I learned a lot about fungi, and molds. Now, if there was one place to go to learn more about UAMH, where would you send?
www.uamh.ca.
Fantastic. I wish all the best for the center. I hope that what little part we can have here in helping it stay open and alive, I hope it has.
I hope so too. Anyway, thanks very much for the invitation.
It was my pleasure.
Yeah.
Thank you.
Great.